Um, hi, my name is Brian, and I'm honored to speak in front of you today. Uh, Thank you for your invitation to let me speak. Once again, my title is An Anatomic Proteomic Atlas of Human Glioblastoma. I am a PhD candidate at the University of Toronto. Um, oh, uh, oh, okay. I'll be going through some stuff. I'll begin with my education. Where did I get started? Some things about my career, and end off with current research. So I went to school at the University of Toronto Scarborough to do my undergraduate degree. I didn't, coming out of high school, I didn't really know what I wanted to do. I know I liked science, but that was pretty much it. My older sister found a program at U of T uh, giving you 2 degrees in 5 years. And I'm like, wow, that's a steal. People usually get 1 degree in 4, 2 in 5? Sounds great. I'll take it. So that's what I did. Uh, I was interested in chemistry. So I pursued a bachelor's of science in chemistry and my bachelor's of education. Um, and really my first step into research was with this professor, my supervisor, Professor Artur Ismailov. And my research was mainly on quantum mechanics and doing molecular mechanics and density functional theory. And again, I didn't really know what I wanted to do. I didn't know what area of chemistry I was interested in. I thought this could be cool. It sounded cool. So I was like, why not? Let's try some undergraduate research. I wanted to try and experience lab. So I tried this. But after I finished my project and working with Professor Ismailov, I knew that this area of research wasn't for me. So after my undergraduate degree, I stopped doing research. I remember that I pursued my bachelor's of education, so I became a teacher. I wanted money. I wanted to travel, so I was like, well, I could make money being a teacher. I could also travel. And there's also this thing called the TOEFL. Uh, TOEFL is this certificate where you train to teach English to than other countries. So a lot of people take this certificate to teach English in Korea, Japan, and China. I guess now it's going to be a lot more difficult, but hey, at the time that was something a lot of people did. But so I tried to do that. I also applied to a bunch of schools in Toronto. Uh, in the end, I didn't go abroad like my uh, dreams wanted me to. I wanted to travel, but I just didn't go. I stayed in Toronto and I got a teaching position at the prestigious University of Toronto Schools. This is a private middle school, high school for some of the most gifted students in Toronto. And I was happy there for a year. I made some money and I was like, I still want to travel. So I quit my job and I went backpacking across Asia. I went to China, Korea, Japan, Taiwan, a lot of random places. And I had a great experience. But at the back of my mind, I knew I still wanted to do science. And teaching is fun, but science was calling me. Research was calling me. So I came back. I came back to Toronto, but I pursued my Master's of Science at York University. Um, from my previous experience in research, I knew that I didn't want to do computational chemistry or quantum mechanics or stuff like that. But I was more interested in analytical chemistry. So analytical chemistry is the science of detection and quantification of molecules. For example, these are the people who are testing if there's lead in your water, or the purity of your pharmaceuticals, or how many calories is in your sausage and egg McMuffin, stuff like that. So quantifying things, identifying things in everything we use and know and love. So I joined the lab of K.W. Michael Su and Alan C. Hopkinson, who is not pictured here. Michael is he's a high-ranking professor and scientist. He is currently the Vice President Provost of Research and Innovation at Windsor University. But at the time, he was my professor at York University. So he gave me a lot of opportunities to learn about this thing called analytical chemistry, and more specifically, this technology called mass spectrometry. I didn't have a lot of experience like in analytical chemistry, but I knew that mass spectrometry was the gold standard to identifying and quantifying molecules and like things of any kind, really. So this was really my first foot into research, and he helped me win a number of awards, and I was able to publish my first 4 papers, and it was a really great experience. All of my papers revolved around identifying small molecules and their structures using mass spectrometry, identifying them and quantifying them. And honestly, I'm really grateful to my supervisors here. They taught me a lot about science and research and being robust with your methodologies, and they were great mentors. So I finished my Master's of Science, and again, I didn't know what I wanted to do with my life. I kind of still don't. but I was like, okay, being a graduate student doesn't pay you a lot, so I want a job. So I got a job. Because of my supervisors, I got a job at Styx. Styx is a $1 billion mass spectrometry company. They're one of the most famous mass spectrometry companies in the world, and its research headquarters are located in Toronto. So that's really good and lucky for me. I was able to land a position there, and again, I was able to work with other different companies, pharmaceutical companies, Pfizer and Merck. They're both $1 billion pharmaceutical companies, and all of my research projects revolved around identifying, separating, and quantifying pharmaceutical drugs. Because of my positions there, I was also able to publish another paper. But I worked there for 1-2 years or so. And again, I kind of realized that I wasn't finished with school. I wasn't done with learning. I had a Master's of Science, but I realized that I could push myself further. So I wanted to do my PhD. I went back to school. I went back to the University of Toronto, and I wanted to try something new. So I pursued a PhD in laboratory medicine and pathobiology. I was still interested in analytical chemistry, but there's just so much more and different things that I wanted to learn and pursue. So one of the things that I was interested in is looking for biomarkers for diseases. So what are biomarkers? Biomarkers are compounds, small molecules that are kind of found in people and that help us to diagnose disease. For example, um, if you're having a heart attack and you're sent to the hospital, one thing that they will, or if you're suspected of having a heart attack and you're sent to the hospital, what they will do is they will take your blood. And within your blood, they will look for this compound called troponin. Troponin is a biomarker that helps the people working at the hospital to know whether you are about to have a heart attack. So depending on the abundance of your troponin levels, we can then say, oh, he's about to have a heart attack. We need to take X, Y, Z procedures. So I thought this was a very interesting concept and something that I would like to pursue. So that is why I chose U of T and this program specifically. And this is where I still am at and where I'm currently doing my research. So I guess from the first half of my presentation, I wanted to give you some take-home messages. Have an open mind. Like, I didn't know what I wanted to do throughout most of my academic, industrial, random things career. I just kind of meandered along and I just pursued what I liked. I always try to explore the world and gain different perspectives, exploring different areas of research and different possibilities and different things that I just thought were interesting. And 3, it doesn't matter how long it takes, you should follow your dreams. I think nowadays there's a lot of push for people to kind of have this straightforward path where after high school you go to university and then after university you pursue even higher levels of education. But I think that's not always true, like especially now, and it's not the best path, certainly not. And I can give you many examples of my friends and clearly myself that don't take the straight path and kind of meander along, and I think it's important so that you can figure out what you really like. For example, I know a friend who did biology in undergraduate. He graduated, he didn't like biology, and he completely switched paths and had to do all the restudying to do law. I know another friend who pursued math in his undergrad, realized he didn't like it when he graduated, and he restudied and pursued history, and now he's a professor of history at Berkeley. I have a friend who did business in their undergrad, who started working at a firm, realized it wasn't for them, came back to study science. So you shouldn't try to pursue a straightforward path. I think you should try to meander a little bit and try and really find what you really like. Sometimes if you try and like follow this kind of rigid mentality, you might find yourself doing something that you don't really like. And when you do that, you won't find as much fulfillment with your life. Okay, so that's what I have to say about my career and my education. And here is some of the research that I'm doing. So my research is an anatomic proteomic atlas of human glioblastoma. So glioblastoma is a deadly brain tumor, most prevalent in adults, and has a median survival of 15 months despite chemo and radiotherapy. So here, this is a cross-section of glioblastoma, and I wanted to show you this because I think it really highlights what makes this disease so deadly, and like explains why people really only survive 15 months. So here we have a cluster of tumor cells. They are dividing rapidly, and they are secreting factors that promote the growth of new blood vessels. And these new blood vessels help to deliver oxygen, nutrients, to really increase the growth rate of the tumor and to help maintain the tumor survival. So the tumor is very smart. But these like newly formed blood vessels, they tend to be weak and they tend to fail. And this leads to these regions called palisading cells around necrosis. So here, because these blood vessels fail, then it leads to these areas where there's no oxygen, so these cells start to die. And while this seems like a good thing, cells also start to move away from this area of low oxygen so that they can survive. And in this process, they gain the unique property of chemotherapy resistance. So now we have a chemotherapy resistant tumor. So it's increasing its growth and it's giving itself chemotherapy resistance. And well, another reason why this disease is so deadly is that it also infiltrates into your normal brain. So it's kind of merging with your normal brain, and we can't remove all of the tumor without also removing large parts of your brain. So this tumor, this disease is extremely deadly. So what can we do about it? Well, the traditional approach to kind of this glioblastoma disease, or to find ways to treating it, is we would take the whole tumor and we would kind of do some kind of profiling, do some kind of analysis, and hopefully develop a drug that can target all of these different areas of glioblastoma. And what we know now is that this kind of methodology just doesn't work. There have been no improvements to the survival of glioblastoma patients in the past 40 years. So clearly some new approach needs to be done. We can't use this one drug fits all approach, and one drug just can't target all of these different like unique properties of glioblastoma. So what can we do? So our approach is we want to develop drugs that can target each of these different properties of glioblastoma. So one that can hopefully stop these blood vessels. So if we can stop blood vessels, then hopefully we can stop the growth of this tumor. Perhaps we can develop a drug against this kind of chemo-resistant area and kind of stop this chemo resistance. Or we can develop a drug that can stop brain infiltration so that surgeons can remove larger sections of this tumor without also removing large sections of the brain. And by combining these different drugs together, we can then target this disease. But towards this goal, we need to be able to understand the biology of these different aspects of glioblastoma. So towards this goal, recently there was a paper published called an Anatomic Transcriptional Atlas of Human Glioblastoma, and it was funded by Microsoft and the Ben and Catherine Ivy Foundation, and Microsoft and its co-founder, Paul Allen. So in this project, they wanted to look at these different regions of glioblastoma that I talked about before and try and find transcription factors, or these RNA, which are the building block, which is kind of the blueprints for all of the proteins or the molecular machinery that are within your cell. So they were able to find a specific RNA that have higher abundance in all of these different areas that I talked about before. But they focused on RNA. These are the set of instructions that make proteins. But RNA doesn't do anything except for make proteins. What is really the function of the cell, and which really does almost everything in the cell, is proteins. Proteins are the molecular machinery that drives nearly all of the function within a cell. So we want to take a complementary approach to this transcriptional atlas and create a proteomic atlas. So we want to find a protein that is upregulated in our tumor area, a protein that is upregulated in these blood vessels, so that we can target these proteins with specific drugs. And then hopefully by combining these drugs together that can target these specific proteins within these different areas, we can then do better and treat this deadly disease called glioblastoma. So how did we do this study? Well, we recruited glioblastoma patients. We recruited 25 glioblastoma patients from University Health Network and Sherbrooke Hospital. Thank you to these families and these patients for giving up their time and participating in our study. And we did a full biological analysis according to current hospital standards. And we really used these patients and to really fuel our research. So we are really eternally grateful. But to really have a robust selection process of these different areas of glioblastoma, we used two kinds of neuropathologists, which helped to identify these different regions that I talked about before, and they were independently annotated and circled, so that only the areas that were agreed upon by these two different neuropathologists, then we were like, okay, since they both agree, then this is the most robust way forward, and then we can look into profiling these different regions. And to profile these different regions and find proteins that are within these regions, we use this this method called laser capture microdissection. And this method takes a laser and physically cuts out portions of the tumor that I was describing before. So here, these blue arrows are pointing to blood vessels. So we want to look for proteins within these blood vessels. And to do that we are going to cut out these blood vessels using a laser. So here's the before, here's the after. After the laser, you can see that this kind of really neat outline of where the blood vessel used to be. And I have this short video of what it kind of looks like. So that's a glioblastoma tumor. Encircled in red is the blood vessels. And here, that's the laser in blue. And where there used to be blood vessels, there's now no longer any blood vessels, as they are removed. And now we are collecting them into a test tube, and we will take that to analyze for specific proteins. Yep. So now we want to use what I know and love, mass spectrometry, to kind of identify proteins of interest within our specific regions. So here are some of my results. So here, this is a principal component analysis. So this is kind of a way to see your samples and how they are separated and how they are unique. So for example, here, we have a lot of normal brain samples. So this is no tumor. Here in red, we have this region, all these samples came from this the tumor infiltrating into the brain, and we can see that they're all clustered together, so that they're very similar as expected. And here in blue, we have only tumor samples. So this gave us a lot of confidence because we got this kind of patterning where we went from tumor only to tumor infiltrating into the brain to only the brain. So this is kind of a safety check to kind of make sure that we are on the right track and what we're doing is correct. So then we did this thing called differential expression analysis to kind of find these biomarkers or these proteins that are specific to these different regions. So we were able to find these proteins that were found in each of these regions. And now we are looking into targeting them with different drugs and seeing if we can improve the survival of these glioblastoma patients. But that kind of isn't it. We also wanted to do more for the community. So we created a website here. No. Is it working? Do you see what website?
Yeah.
Oh, okay. So here, we wanted to make this resource for the community. We wanted to allow researchers from around the world to kind of study what we did, analyze it, and hopefully be of some use in the future. So we created this website called brainproteinatlas.org. Here we have all of our patients and their kind of clinical history. And what this website does is it allows for bioinformatic analysis of these different regions that I talked about before. So here we have this like bulk tumor, cellular tumor mass, compared to the infiltrating tumor cells into the normal brain. And we can see a number of different proteins that are statistically significant when you compare one region to another. And we have all of that information here, and you can do a lot of cool bioinformatic analysis, and hopefully researchers around the world will find it somewhat useful. We also integrated a scatter plot, so you can see the relationship of one protein to another protein. So you can see that this protein, when you plot this protein and this protein, you can see that they kind of increase in relationship together. So there's this positive correlation. When you have an increase of this protein, you also have an increase in this protein. And this has a number of biological implications, as there's clearly some kind of a relationship between these proteins, and hopefully it can fuel more discovery, more research, and so we can target this deadly disease. We also wanted people to be able to look at the abundance. So here we have, here people can look up a protein of interest like that, and then they can see what is the relationship of this protein like within these different groupings that I talked about before. And we can see that it's upregulated in our blood vessels. And hopefully that this can be of some use to people in the future. Okay, and let me go back to the presentation, if I can find it. Okay. Oh. Again, share. Okay, so back to the presentation. So again, we created this this kind of publicly available online database to kind of fuel future research. But that's basically the end of my presentation. But like a lot of research that happens, no one does it alone. So I would like to thank all of these people who helped me in my journey and helped me to do some of this research. And most importantly, I want to thank the patients and their families for their tissues, their donations, and their time, because truly without them, this work would not be possible. So that's really my presentation. So
Thanks. Very well done.
go for the questions
So before
now.
we get to the questions, I just want to say thanks so much for that presentation, Brian. I really enjoyed it. The research, of course, very cool, but also the career side. I myself am at a bit of a career crossroads, so it's always nice to hear some other people's stories.
Yeah.
So while I invite the audience to submit their questions for Brian into the chat, and while I'm sort of waiting for those to roll in, I'm going to ask a couple myself. So it's probably pretty likely that most of the audience has heard of mass spectrometry before. But
Okay.
just in case people are not quite up to speed, would you mind just giving us a little kind of basic rundown of the technology and sort of how it's applied in biological systems and specifically proteomics, and also maybe tell us what a proteome is?
Sure. Mass spectrometry is is a technology that separates out compounds based on their mass. So by identifying, like by separating compounds or molecules or proteins or really anything based on their mass, we can hopefully then identify and quantify them using their mass. And what is a proteome? The proteome is basically all of the proteins that make up a cell. So for a cell to function, to do anything, it needs to make proteins. Proteins are the molecular machinery that makes a cell a cell. So a cell needs to make specific proteins to do a function. So a blood, so an endothelial cell or a blood vessel cell uh, makes certain proteins that are different from those that are experiencing, say, a cell that is infiltrating into the normal brain tissue. And these differences in proteins and their abundances is what makes each cell unique, and which, like, hopefully, and which, like, using mass spectrometry allows us to quantify and identify these differences. Is there any other?
Oh yeah, makes sense.
I
I want to ask one more question before I get to some of the submitted ones.
Sure.
So um, something that I think a lot of people don't know about is how much research that goes on is in time, how much time is spent in developing these sort of free online resources that are open to everybody. So I think it's really cool that your group is sort of part of that world. And just in case the audience might not know, this means that that group's, website is sort of available to all kinds of other researchers to refer to and use to help with their own research. Could you just talk a little bit about like what that is and sort of how you might interact with other research groups that are using your database,
Yeah.
and how important that is for the sort of opus of science?
Yeah. So definitely, like online databases and these like publicly available websites are becoming more and more of a trend, mostly because like it allows other researchers to kind of probe your work, so that then it's just like known to be more robust. Because like if you allow free and access probing, then it's okay, you clearly have some confidence in your work. And it's also like it allows other researchers to like study your work and really expand the field. So I'm also, the importance is you can look at the Ivy Gap, right? Um, Clearly, they did a very similar thing to what we did, and they have a free and online open source website powered by Microsoft and the Ben and Catherine Ivy Foundation. It costed them like $10 million, and they have 10,000 unique users per day, no, per month.
Wow.
So that's a lot of people and a lot of money. So these things aren't like cheap, they aren't easy, they take a lot of time and effort, but I think that these online resources are kind of the way of the future, and it's a lot of people use them, and yeah, it, it's like it's really helping to fuel science and fuel other researchers.
Yeah, thanks. I myself refer to these a lot, so it's
Yeah.
interesting to meet someone who makes them. Okay, well, let's get to some audience questions. So Alice asks, how early would you recommend getting involved in research? Is the first year of university too early?
So I actually um, I don't know how to answer that question, to be honest. So I can talk about my experience. I never did that. A lot of people do. So in I again didn't know what I wanted to do with my life. In first year, no idea. I just took my courses, and that was it. I didn't apply for co-op. I didn't apply to labs. I didn't do anything. Um, I didn't do it in my second year either, or my third. I, I basically only started in my fourth year. I don't think it's too early. I know that even people in high school like apply to labs. I know a number of high school students who like apply to labs and like try and do research with them. Like perhaps it may not be the most like intensive, but I think if you're interested in something, pursue it. Like there shouldn't be something in your mind saying, oh, I'm not ready for this. Oh, like you should kind of have this like positive outlook and just try. Like, what's the worst that can happen?
Nice. So Hanem asks, why does the dying of cells around the newly formed blood vessels cause chemotherapeutic resistance?
Oh, so without getting too technical, so it kind of like so it upregulates a pathway, like a hypoxia, or it's called like low oxygen pathway, and basically through this really complicated network of low oxygen, the cell responds to it by creating these new proteins, and a side effect of that is that it somehow blocks these chemotherapy agents. Yeah, that's a really basic answer, but I can link you like to some papers online, but that's basically the way I would say it, or that will that will link you to da, da, da. Sorry, I'm doing something right now. Wait, yeah. Uh, how about we do this? Where's my thing? Where is my thing? Google Chrome. Where is my desk? Oh, here. So read this paper if you can. Yeah, and it talks about it. I wrote it. Yeah. And it's free. Then it's online. You can just click. You don't need to pay anything.
Nice.
Yeah.
He says, thank you so much. Um, okay, Alex asks, Are you also developing protein interaction graphs or interactomes for glioblastoma?
So no, uh, I don't have much interest in the interacting networks of proteins, but there are definitely like other researchers who are. Uh, I think. Okay, clearly I might not have spelled his name wrong. No, yeah, this guy does. Yeah, so this guy does like a lot of work into like protein interaction networks and using mass spectrometry to study diseases. I personally am not interested in that, but other people are. Look at some of his work.
Cool, thanks. Manisha asks, oh, Alex says thanks. Manisha asks, what are some tasks that graduate students and researchers must do other than research?
Um, this. Technically, this isn't research, so I think this qualifies. But there's really no tasks that you need to do. Maybe there are tasks that you want to do. We're still humans, so you still like to do like non-work related things, but need to do other than research.
I don't know,
Eat.
maybe being when you're, participating as part of a research group, administrative things, housekeeping, etc.
Oh, okay.
Doing seminars.
Okay. So, so some things along those lines. Um, okay. So it depends on your group. Um, sometimes say if you're like an undergraduate researcher or something like that, then you are kind of involved in like more administrative tasks. Something that's kind of outside research is that PhD students tend to take on undergraduates. So usually undergraduates who apply to the group are like instead of working directly with the like head boss guy who usually doesn't have time, and you usually work alongside like a PhD student to kind of guide you along your way. Um, Other housekeeping things. I think something else that you need to do is you should kind of try and get along with your lab mates. I think this is really important and kind of like networking. Um, uh, Talk to the people around you, see who are the big names in your field, what you're interested in doing, and talk to them. Like at the end of the day, we all like work together and like trying to have the same common goal. But to do so, like you kind of want to like mingle and like get to know your coworkers. I guess. And it also helps with your career in the future. The only reason I got the Styx job, uh, the company I worked for, was because my supervisor, Michael Su, his first student was the principal scientist, so the head honcho boss guy at Styx, which then I was like, hey, I'm also a student there. Hire me, please. Yeah, so networking.
Cool, thanks. Okay, Hanem is back with another question. How do you recommend contacting labs to start working with them?
Email. So here, I have a thing. Um, Take this email and email them, but don't just write an email that says, "Oh, I am so and so. I would like to work in your lab. Please and thanks." What you should do is read this. Read all of the research that they're doing. Look at their publications. Like find like a publication that you think is really interesting, and then you should be like, oh, I thought it was really cool that you did this and this in this paper, and looking at this, I think it's really meaningful towards the field of so and so, and that I would really like to learn more about these technologies and the science behind whatever the person's studying.
Right. Kathleen asks, is it possible to get involved in writing papers without a PhD? She adds, for example, could you be hired to write a paper for someone else's research?
Yes. Like, so there are definitely people who just write papers. Um, like, so, like, it really depends on the person and the lab, but I know of of here. Let me see. Diamandis Lab 2. I actually don't really know our lab website. I think it's diamandislabii.org. Might be. Oh, wait. Okay, let's see if this is an actual website. Nope, it's not. Okay, where is it? Maybe it's on his, okay, here. Yeah, so team, let me scroll down. Let me see. Yeah, so like, so Jessica and Claire, they basically volunteer and they just write grants, so grants and papers. So that's all they do. They don't actually do any research, and they just write for us. So that's definitely something that you can do if you're interested in. Yeah, why not?
Cool. Okay, so the audience, we're about to finish up here. So if anybody has any more last minute questions, try to get them in now. I'm going to go ahead and ask one more while we're waiting. Can you tell us sort of what a typical day looks like for you when you're working?
Oh, okay. So I think it kind of changes, but um, so I'll give you 2 days. One like COVID day and like one like pre-COVID day
Perfect.
to to give you like a better idea. So I guess a COVID day, I would wake up, I go on my computer. No. Okay, I lied. Okay, so I wake up, I go to the gym, then I come back, I go on my computer, I read emails, and I work on some kind of bioinformatics stuff. Can I show you? Let me see if I actually have something. Da, da, da. What? Let's see if I can do something. New share. Da, da, da. Sorry, desktop. Okay, this one. Share. But do you see my screen? But I
Yeah.
can't actually tell. Okay. So uh, so I use like R a lot. I use R. I use a bunch of different programs and doing bioinformatic analysis and stuff. So I'm usually just on the computer all day. But I also go into lab. Our lab opened a couple months ago. But we have we have strict guidelines. We are only at 50% capacity, and we are in specifically designated groups, and we get tested regularly and stuff. And when I'm in lab, we don't really talk to. I don't really talk to anyone because you can't really, and you can't really stay around. But I just do some lab work or some bench work, maybe growing some cells, um, and I could go home. Yeah, it's not much during these COVID periods, I guess. But before COVID, I would do a lot more. Um, we a typical day depends. So I still did a lot of bioinformatic analysis like this. I use a lot of programming and stuff like that. So a lot of my time is on the computer, like compared to other people. Um, yeah, but I would get like more opportunities to interact with my colleagues. I was allowed in lab a lot more, like doing bench work, um, and giving presentations like this. I haven't. The last presentation I've given was a long time ago, and it was in person at Sherbrooke University, and yeah, so it's kind of nice to give new presentations.
Yeah, we're happy to have you. Okay, Emma asks, did you learn to use R before you entered university, or did you learn it along the way?
I I learned it along the way. I most certainly did not take courses in R. Uh, I think I'm fairly proficient, but I basically learned through the internet. I studied. I, I can show you how I did it. Where's I use I use this. Yep. So I used this website called Datacamp, and that's how I learned R. I took all of their projects, their skills set, their training, and I did it in university. I didn't do it beforehand, and most certainly, I don't think that you need to learn R. Like I think that sometimes a lot of people think that, well, usually I think a lot of people are like, oh, R is going to be so important, or oh, you need to learn machine learning and you need to learn AI. But I don't necessarily think so. I think that what is most important is figuring out what you want to learn. Like, I like bioinformatics, I like big data, I like identifying and quantifying like different proteins, different molecules, what have you, right? But not everyone uses R and needs to use R. So a lot of people do a lot of molecular biology work. So they are using CRISPR to knock out a gene and looking at the phenotype. And for that, you don't need R. Like, R is useless for that. Um, like, oh, if you're using uh, like if you're trying to study like some form of like physical chemistry and you want to see the energetics of a molecule, you don't need R for that. Like, so when you try, like, so you shouldn't generalize and like kind of pursue like a technology just because everyone's like, oh my God, it's going to be so important in the future. Figure out what interests you have, and when you understand that, you'll need you you'll then like learn what technologies and what things are important in that field.
I think that's a great answer. I totally agree. Uh, maybe needed to hear it right now. So Emma says, all right, thanks for the advice. So I think we're going to leave it there because we're just about out of time. I just want to start by thanking the audience for being engaged and submitting some really great questions, and most importantly, thanking you, Brian, for lending us some of your time and your expertise and opinions along the way. I think that everybody, myself included, learned a lot. Before I let everyone go,
Oh, oh, uh, Liam?
oh, did I mute? I muted. So all I said was that we'll be taking a 1 hour break in the seminar series starting now. So we'll be starting at 3 p.m. Eastern with Mattia, who's going to be talking about the battery of the inner ear. So I hope that you'll meet me back here. Well, not exactly here, but at the Zoom link that I just posted in the chat in an hour. And otherwise, just thanks again, Brian, and to the audience. Everybody have a good day.
Uh, Bye. Thank you for having me.
Oh, our pleasure. Everybody's saying thanks, Brian, in the chat.